Good morning, everyone. Today, I will discuss the bacterium, Listeria monocytogenes. This presentation will cover its habitat, morphology, cultural characteristics, biochemical characteristics, host range, and virulence factors, as well as its mechanisms of antibiotic resistance. I hope you will find this information useful and informative. Awareness of Listeria monocytogenes, a gram-positive bacterium, is important due to its potential to cause serious illnesses in humans, possibly even severe complications and death. It is commonly found in soil, water, vegetation, and food processing environments, and those with weakened immune systems, such as pregnant women and their fetuses, are at an especially high risk. To prevent illnesses caused by this pathogen, practice appropriate prevention methods. Maintaining good hygiene practices in food processing environments is important in order to minimize the risk of contamination, as Listeria monocytogenes, a bacteria found in a variety of environments, is capable of colonizing both raw and processed foods. Despite its size, Listeria monocytogenes is an anaerobic, gram-positive bacterium that can be identified through its tumbling motility caused by the presence of Paratrigus flagella. It measures 0.5 to 2.0 micrometers in width and 0.5 to 10 micrometers in length, which makes it distinct from its peers and allows for further study. Listeria monocytogenes, with a ground glass appearance when grown between 30 and 37 degrees Celsius, has a wide temperature range for growth, from 0 to 45 degrees Celsius, making it difficult to contain. Identifying and controlling it is essential to avoiding foodborne outbreaks. Gram-positive, facultative anaerobic, rod-shaped bacterium Listeria monocytogenes possesses a number of biochemical characteristics. It is catalase positive and oxidase negative, able to ferment glucose and other sugars and hydrolyze esculin and lecithin. Moreover, it produces phospholipases, which can contribute to its pathogenicity. Listeria monocytogenes is a bacterium that is known to infect many types of animals, including humans, cows, pigs, and birds. With its wide host range and ability to infect various cell types such as epithelial cells, macrophages, and dendritic cells, this bacterium is a particularly versatile pathogen. Listeria monocytogenes produces a range of virulence factors, including internalin, Listeriosis O, and several phospholipases. These factors enable the bacteria to invade and replicate within host cells and avoid the host's immune system. Fortunately, Listeria monocytogenes is generally susceptible to antibiotics like ampicillin, trimethoprim sulfamethoxazole, and erythromycin. Early diagnosis and appropriate treatment can typically stop the progression of serious illnesses. Listeria monocytogenes is a bacterium that is usually susceptible to a variety of antibiotics, such as ampicillin, trimethoprim sulfamethoxazole, and erythromycin. However, some strains of this bacteria have developed resistance to certain antibiotics, posing a challenge for treatment. As such, alternative therapeutic methods may need to be employed in order to address this resistance. Listeria monocytogenes is a foodborne pathogen that can cause severe infections, particularly in people with weakened immune systems. Studies have revealed that this pathogen is mostly discovered in polluted deli meats, soft cheeses, and unpasteurized dairy products. Research has specified that some populations are at a greater risk of infection, such as pregnant women, the elderly, and those with weakened immune systems. To decrease the potential of exposure, it is essential to recognize the potential sources of Listeria monocytogenes. Listeria monocytogenes is a foodborne pathogen capable of entering our cells and replicating within them, potentially leading to tissue damage, stimulating an immune response, and cell-to-cell transmission. Without proper treatment, it can cause severe conditions such as sepsis, meningitis, and other fatal diseases. Therefore, precautionary measures should be taken to guard against the risks of infection from this pathogen. Listeria monocytogenes is a bacterium that can cause food poisoning. Once ingested by the host, it is recognized by the innate immune system and triggers signaling pathways which result in the production of cytokines and chemokines, which in turn attracts immune cells such as neutrophils and macrophages. These cells then phagocytose the bacteria and use various mechanisms to kill them. Comprehending these immunological responses is vital for controlling the spread of this bacteria. Listeria monocytogenes is a bacterial species that can lead to infection in humans if consumed. Detection of this species is typically made through culture of clinical specimens of infected patients or through molecular methods such as PCR, which involves isolating the bacterial DNA and identifying the presence of Listeria. To prevent infection, it is important to consume only properly cooked food, avoid unpasteurized dairy, and practice stringent hygiene standards. Proper food handling and preparation is paramount in preventing Listeria monocytogenes infection. This includes cooking raw foods to the appropriate temperatures and avoiding high-risk foods such as deli meats and unpasteurized dairy products. Furthermore, pregnant women and other individuals at high risk should take extra measures to avoid potential sources of contamination. Listeriosis is a serious infection caused by the bacterium Listeria monocytogenes and is primarily treated with antibiotics such as ampicillin or trimethoprim sulfamethoxazole. In severe cases, hospitalization may be necessary to manage complications such as sepsis or meningitis. Symptoms associated with listeriosis include fever, muscle aches, nausea, and diarrhea. Early diagnosis and treatment are important to preventing further complications, so it is important to be aware of these symptoms. Thank you for taking the time to listen to this presentation, and I hope it was helpful.